Spermatogenesis, the birth of the male gamete. Hi there! Here are a few thought-provoking questions for you. What are the advantages of sexual reproduction over asexual reproduction? How does the exchange of genetic material during sexual reproduction contribute to the survival of species? What might happen if gametogenesis did not occur or was impaired? Let's get into it. Trust me, this will be so interesting, you don't want to miss it. Addressing our very first question, sexual reproduction has its benefits. It's like a genetic mixtape mashup that brings together genes from two parents, like a DJ mixing beats. This wild genetic party creates offspring with greater diversity. Just like a mixtape with different genres, genetic diversity helps populations adapt to changing environments, increasing their chances of survival. Another advantage is that when genes do the tango during sexual reproduction, they engage in a move called recombination that helps eliminate harmful mutations through natural selection. It's like a dance-off where only the best moves make the cut. This way, species can weed out any mutations and keep the dance floor rocking with healthier genes. Let's address our next question. A great example would be that of bacteria and viruses, like an evolution on the go. Bacteria and viruses throw the wildest genetic raves. Sexual recombination in these tiny creatures allows them to shuffle and exchange genes like crazy. This rapid evolution helps them adapt to new challenges, like developing resistance to antibiotics or finding sneaky ways to dodge the host immune system. Didn't we all witness this during the massive COVID period? The virus mutated way quicker than we could handle. Another great example is the cross-pollination in plants. Yes, that's right. Plants know how to get their genetic groove on too. Through cross-pollination, different plants come together for a jam session of gene mixing. This creates incredible genetic diversity among plant offspring, making them more resistant to pests, diseases, and environmental stressors. Now think about this. What if gametogenesis hit a sour note? Well, if gametogenesis didn't happen normally, there would be no formation of sperm and eggs. That means no sexual reproduction. And the dance party would come to a halt. Species would miss out on the benefits of genetic diversity, making them less adaptable and more vulnerable to extinction. Impairments in gametogenesis can also lead to fertility issues. Let us now define gametes. Gametes are the super specialized reproductive cells involved in the ultimate concert called sexual reproduction. In humans, we have the cool dudes called sperm cells in males and the fabulous divas called egg cells in females. The process of their production is called gametogenesis. What makes gametes extra special is that they are haploid cells, that is, they have half the number of chromosomes compared to other cells. Are you wondering why gametes need to have half the number of chromosomes compared to other cells? This is because the gametes have a mission to keep the chromosome party balanced. By having half the number of chromosomes, they ensure that when they unite during fertilization, the party doesn't get too crazy with too many chromosomes. They know the zygote needs the perfect diploid number 46. Plus, having different combinations of chromosomes from two parents gives the offspring a chance to shine with their own unique style and traits. It's like getting a blend of awesome features from both mom and dad, creating a one-of-a-kind star. Did you know? Humans aren't the only one throwing a genetic party. Other organisms have their own gamete shindigs too. Plants have their plant-style gametes like pollen and egg cells, making sure they can reproduce and bloom. In the animal kingdom, you can find gametes like sperm cells in dogs, cats, birds, and fish. Fungi and protists have spores as their gametes. Alright, let us move on to classify gametogenesis. Spermatogenesis is the specific form of gametogenesis that occurs in males, resulting in the production of mature sperm cells, while oogenesis is the specific form of gametogenesis that occurs in females, leading to the production of mature egg cells or ova. Let us now learn about spermatogenesis. Spermatogenesis begins in males during puberty, when hormonal changes stimulate the development of the testes and the production of sperm cells. This complex process takes place within the testes of male individuals, specifically the seminiferous tubules. There are many steps involved in this process. First up is the development of the testes. During embryonic development, the gonads, or the primitive reproductive organs, initially have the potential to develop into either testes or ovaries. In males, a gene called the SRY gene on the Y chromosome triggers the development of testes from the genital ridges located in the abdominal region of the embryo. The next step is the formation of testicular cords. As development progresses, the testes begin to differentiate and undergo morphological changes. Specialized cells called Sertoli cells start to form testicular cords within the testes. Meanwhile, primordial germ cells migrate from their site of origin in the yolk sac towards the genital ridges and settle within the testicular cords. These are the precursor cells that will eventually give rise to sperm cells. The next step is the differentiation of Sertoli cells. The Sertoli cell play a crucial role in supporting and nurturing the developing germ cells and secrete factors necessary for their growth and maturation. And now, the actual action is meiosis, the process of cell division that produces haploid cells with half the chromosome number, that is, 23 chromosomes. In spermatogenesis, it occurs within the seminiferous tubules of the testes. Two successive rounds of meiotic divisions take place, resulting in the formation of haploid cells called spermatids. The final stage of spermatogenesis is called spermiogenesis, where the spermatids undergo significant structural and morphological changes to become mature sperm cells. During this process, the spermatids develop a distinct head, midpiece, and tail. The head contains the nucleus, which carries the genetic material or DNA of the future offspring. The midpiece is rich in mitochondria, providing the energy needed for sperm motility. The tail, also known as the flagellum, propels the sperm forward, aiding in their movement toward the egg during fertilization. The curtain closer is the release of mature sperm. Once spermiogenesis is complete, the mature sperm cells detach from the Sertoli cells and enter the lumen of the seminiferous tubules. From there, they are transported through the rete testis, epididymis, vas deferens, and ultimately the urethra, where they can be ejaculated during sexual activity. And that is the transformation of primordial germ cells into fully functional sperm, ready to continue on their journey of fertilization, which we will discuss in the following sessions. Let's discuss what an axoneme is. The cytoskeleton of cilia or flagella is called its axoneme. In the context of spermatogenesis, the axoneme is a specialized structure within the sperm tail that plays a critical role in sperm motility. The axoneme structure and the coordinated movement it enables is essential for the sperm to propel itself through the female reproductive tract toward the egg. Without a functional axoneme, the sperm would be immotile and unable to reach its destination for successful fertilization. Pop quiz. The entire process of spermatogenesis is initiated and monitored closely by hormones. Here are some of them that run the show. First, we have the follicle-stimulating hormone, or FSH, that acts as the conductor of the dance, guiding the other hormones and setting the tempo. It stimulates the Sertoli cells, the lead dancers within the seminiferous tubules, to provide essential support and nourishment to the developing sperm cells, helping them learn their moves. The next character is the luteinizing hormone that shouts, "Lights, camera, action!" It acts on the latex cells, which are like the production crew behind the scenes, and prompts them to produce testosterone, the hormone responsible for masculine traits. Now, coming to the main act, testosterone. This hormone shines as the primary male hormone produced by the latex cells. It not only enhances the growth and maturation of the sperm cells, but also adds a touch of masculinity to their performance. Testosterone gives the sperm cells their distinctive features, making them strong, agile, and ready to impress. We then have the gonadotropin-releasing hormone, which is like the choreographer, working behind the scenes to ensure the dance routine flows seamlessly. It originates from the hypothalamus and directs the anterior pituitary gland to produce FSH and LH, making sure they are in sync. And finally, the hormone inhibin acts like a manager backstage, without which there would be utter chaos. Inhibin is produced by the Sertoli cells and acts as a feedback mechanism to maintain a balance. When the dance routine gets too intense and FSH levels rise, the hormone inhibin steps in to regulate the production of FSH, ensuring that everything stays in control. With that, we come to the end of this session of spermatogenesis. In the next session, we will study the similar process of gamete formation in females, a process called oogenesis. We hope you had fun learning with us.